Hi. Welcome everybody to the second to last class. You guys have stuck it out, and today we're going to be talking about, we kind of have blended the biology and clinical aspects. Oh, could somebody go on mute there? We've been kind of blending the biological and clinical aspects with the informatics in some of the last slide decks. And today what we want to do is turn our focus just to the informatics aspects of genetics. And a lot of it is going to be a recap of what we've covered before. Maybe one of you guys could go on mute out there.
How to do the muting during.
On your computer, put your cursor up to the top, and on the left, pops down.
Okay.
All right. Thanks, Dan. Okay. So today it's clinical genetics informatics. Tomorrow are the ethical issues in genetics. Just a reminder that next week, when we talk about ethical issues in genetics, there's no didactic for that session. It's really what you guys bring. So we'll see. The debates could go the entire time. It might be a short class. It just depends on how actively you guys want to participate in that one. And then that will be it. So today, we're going to go through some questions. We're going to review the homework from 2 weeks ago. And mainly what I wanted to do today was provide a recap of the bioinformatics resources and challenges we've encountered throughout the course. Each week as we present a case and then we look at various types of scenarios, we look at various types of scenarios where we're trying to ping maybe a knowledge source, or we're trying to address some type of clinical use case. We bump up against informatics resources, we bump up against informatics issues, we bump up against informatics challenges. And one of the things I wanted to do today was pull all of that stuff together into one deck. And so much of the meaty portion of today is going to be a recap. And that's why I kind of am not sure how long it's going to take to go through the material. I don't want to kind of walk through everything again. I think that would be kind of boring. On the other hand, having all the material compiled together in one deck, I'm hoping will be a valuable resource for you guys. You'll see that all of the informatics sites, tools, and references that we've covered throughout the course have all been compiled into this deck. So we'll go through a clinical case of hypertrophic cardiomyopathy, then we'll do the recap. Then I tried to come up with a very complex clinical scenario, a real-world clinical scenario that would really, push the envelope of bioinformatics tools, public knowledge bases, and standards. And the scenario we're going to be walking through today is a reanalysis scenario. And this will be a patient that has hypertrophic cardiomyopathy where he had next generation sequencing. They found some variants of uncertain significance, but a year later, knowledge has changed. And as a result, there's interest in reanalyzing his existing DNA store. And these reanalysis scenarios, they're fairly complex, and they really do exercise a broad range of bioinformatics standards. So I thought it might be nice to try to pull in a lot of the informatics standards through a clinical case, and that case will be hypertrophic cardiomyopathy. So we'll introduce the case, then we'll do a little bit of a recap, and then when we get back to the case, that's going to be, I think, a fairly complex scenario that might help illustrate how a broad range of standards can be used in a complementary fashion. And then normally we don't walk through the references, but today we'll spend a little bit of time walking through the references so you can see what all has been compiled together for you in this deck. And I think also normally the class is a bit more didactic than interactive. I think when we go through the recap today, my plan is to not spend a lot of time rehashing stuff that we've covered in prior sessions. The intent here was really to kind of pull everything together so that you have everything in one place. But by all means, as we're going through the recap, if there are aspects that you do want to focus on in a little bit more detail, and you want to refresh yourself on what those were, we certainly can talk about these things in more detail. Okay. So just a reminder, next week for the ethics class, we're going to be debating 2 questions. And again, there's not going to be any didactic. I may bring a case that kind of illustrates an ethical dilemma. But that'll just be kind of a launching point. And then from there, we'll open up the debate, which we won't be recording. And 2 weeks ago, we had a homework assignment, and I wanted to go over that before we dig into today's material. And so in the homework assignment, you were given 4 different variants. And for each variant, we were going to try to figure out what is the gene in question, is the variant pathogenic, what disease, if any, is the variant associated with, and where did you find your information. And the responses were very thoughtful and actually illustrated to me how difficult it is to really establish the pathogenicity of a variant, because we got conflicting information from the people that turned in their homework assignments regarding pathogenicity, with some pretty good arguments to support their claims. So we can debate this if you'd like, but this is my best take on the homework assignment. The first variant was found in ClinVar, so that was pretty straightforward. The preponderance of evidence in ClinVar says that this is a pathogenic variant in the GALT gene that's associated with this particular disease. And everyone was in concurrence on this one. This one here, there was some slight variability in the homework responses. This was also a variant that was found in ClinVar, and it was felt to be likely benign. The question here, what disease is this associated with? You know, it's not meant to be a trick question, but the question is, you know, some of you said that this was associated with beta thalassemia, but on the other hand, it's a likely benign variation. So, you know, is it really correct to say that this particular variant is associated with a disease when it's a likely benign variant? So at least here, I've said no, it's not associated with any disease because it's a benign variant. But I understand, you know, when you look in ClinVar, it shows that there's an association with beta thalassemia. And so some of you said, well, then the disease it's associated with is beta thalassemia. Then the last 2 started to get progressively harder. This particular variant here was not present in ClinVar or dbSNP. in order to research this particular variant, you'd have to go hunting around for a locus-specific database where you could find information about it. And so I found information about this particular variant. And at that particular site, it did show a phenotypic association with Duchenne's muscular dystrophy. However, the website doesn't include pathogenicity information. On the other hand, if you look at this particular variation, it's a nonsense mutation that changes the fourth protein, a tryptophan, to a stop codon. So it would be pretty severely damaging to the dystrophin protein. And so based on the ACMG guidelines for determining pathogenicity, even though pathogenicity wasn't stated at the site, I concluded that this was a pathogenic variant. And that was pretty tricky because you had to find information about this variant at a locus-specific site, but then you also had to go try to apply the guidelines to see if you could calculate, based on the ACMG guidelines, whether or not this was a pathogenic variant. And in this particular case, because it's a nonsense mutation, I think we could safely say that this was a pathogenic mutation. And then the last one I thought was particularly tricky. The first thing to do when you have the FIRE resource is to try to come up with a corresponding HGVS syntax, and I've shown that here, that you can then use to search. And this particular spot in chromosome 9, there's no known gene at this location. This is a spot that's kind of in between genes. But this particular SNP, the change from a G to a C, has been associated with an increased risk of coronary disease. And you do find this particular variant in DB-SNP database. So it's possibly associated with an increased risk of coronary disease. It's a very common variant. In some of the population studies, we see this variant, 40, 50% of the population have this particular variant. And so the question is, how do you label this with respect to pathogenicity? And so here there was really the greatest variability in the respondents to the homework assignment. Here I simply called it uncertain significance. And one of the challenges we have here is that the ACMG guidelines for labeling a variant as pathogenic or not really only apply to Mendelian variations. And so here, this is a variation that's not inherited in a Mendelian fashion. This would be part of a constellation of changes that might affect someone's risk. And so really, it's unclear, and there's no clear guidelines out there about how do you label a SNP that puts you at increased risk. So everyone got full credit for this one, but I called it uncertain significance because we don't really have, at least from what I could find, we don't really have a good way of labeling a variant with respect to pathogenicity when it's an at-risk allele as opposed to an allele that's responsible for a Mendelian disorder. So any comments? Yeah.
Yeah, so trying to put together the pieces, it sounds like there is a statistical phenotypic association, but absolutely no understanding mechanistically of how the mutation would be associated with that risk. Is that correct?
Yeah, that's correct. And maybe to even push it a little bit farther, sometimes when you see a statistical association between a SNP and a disease, it's not necessarily the case that the SNP is anything more than just next to the region, and that because of linkage disequilibrium, that particular SNP just happens to be one of the SNPs that we're testing, and it just happens to be next to a pathogenic SNP. So we know that there's a statistical association between this particular SNP and the disease, but we don't know anything really about the pathophysiology, the pathogenesis. And we don't know with 100% certainty that this particular locus is any more than just associated through linkage disequilibrium.
Okay, thank you.
Mm-hmm. So any other comments, debates? This was really a tricky one, and I think the responses from the homework were really very thoughtful, and to me were more illustrative of how hard it is to really kind of label certain variants as pathogenic or not. You guys all got A's, by the way. Okay, so that's the homework. Now we're shifting to our case of hypertrophic cardiomyopathy. And here we see, well, we kind of, obviously these pictures are a compilation of pictures to illustrate the case. But our case today, excuse me, is a 30-year-old man that presents to clinic with shortness of breath, palpitations, and chest pain. His father has congestive heart failure, and his brother had sudden cardiac death at the age of 18 while he was practicing football. The cardiologist explained to the patient the possibility of hypertrophic cardiomyopathy running in the family. On exam, his cardiac exam was consistent with an enlarged heart with an apical impulse that was laterally displaced. He had a fourth heart sound present. His jugular venous pulse pressure appeared to be elevated, and that's reflective of the stiffening of the heart. And he had a double carotid arterial pulse. He had an echocardiogram that showed asymmetric septal hypertrophy. And if you can kind of orient yourself to this picture here, we see here's the left ventricle, here's the mitral valve, and here's the left atrium, and here's the aorta coming out here. And this is the interventricular septum right here, and you can see it's markedly thickened. It's much wider than it's supposed to be here. What we also see here would be a biopsy specimen, and one of the things that you see here is significant disarray of the cardiomyocytes. I'll show you a picture of normal cardiomyocytes in a minute, but normally they're more parallel in their orientation. And here we see marked disarray of the cells, and we also see a lot of interstitial fibrosis. On the gross view of the heart, you notice here this markedly hypertrophied interventricular septum bulging into the outflow tract for the aorta. Anyhow, consistent with this patient's clinical history, physical features, and family history, he was felt to have a familial form of hypertrophic cardiomyopathy. And if we look at his family history, we see here that his father had congestive heart failure, and his brother died at the age of 18 from sudden cardiac death. And these cases of hypertrophic cardiomyopathy, roughly half of them are familial, and of those that are familial, the typical inheritance pattern is autosomal dominant. And in a patient like this, where we can see that he's got a positive family history, about 60% of patients will have a specific mutation identified in one of the genes that code for proteins of the sarcomere. or the contractile apparatus of the heart. So we suspect that this patient probably does have a mutation in one of his sarcomere genes. And I thought it might be fun to kind of take ourselves back to high school biology or medical school biology and remind you a little bit about what a cardiomyocyte is and a little bit about the contractile mechanism of the heart. It's a little bit of a digression, but I didn't have a lot of information to cover today, and I thought this would be interesting because once we cover this picture, then when I tell you what the mutations are that we typically see in hypertrophic cardiomyopathy, it'll make a little bit more sense. So over here we see typical cardiomyocytes. We see that they branch, they connect to each other through these intercalated discs. Cardiomyocytes usually have just one nucleus, but they can have several nuclei. And so here we see again cardiomyocytes that are branching and can have one or several nuclei. And you'll see that each one of these cells is really a big bundle of these myofibrils. And these myofibrils are the contractile apparatus of the cell. And what happens is when the cell depolarizes and the electrical current sweeps over the cell, it causes this large influx of calcium into the cell. So now we have a much higher concentration of calcium in the cell. Each one of these myofibrils, if you look more closely, is comprised of these thin bands comprised primarily of actin and these thicker bands that are comprised primarily of myosin. And what happens is when the calcium level rises in the cell, it results in an interaction between these myosin bands and the actin bands. And you can just imagine these actin bands sliding over. This band would slide over to the right, and this band over here would slide over to the left. So the increase in calcium causes the bands to slide, and that results in the contraction. And here we see the thin actin band, which is also comprised of troponins, cardiac troponins, and tropomyosin. And here we see the myosin band. So these cardiomyocytes, the contractile mechanism, is really based on actin and myosin interactions. And this is primarily where we see mutations in patients with familial hypertrophic cardiomyopathy. These are particularly concentrated in actin, myosin, and troponins. And here's a zoom in of the cardiac sarcomere. A sarcomere, by the way, is the space between one of these Z lines and another one of these Z lines. It's the functional unit of contraction. And if we zoom in on one sarcomere, we'll see here the thick band of myosin and the thinner bands of actin, along with tropomyosin and troponins. And the most common mutations that we see here are mutations with the beta-myosin heavy chain and the myosin-binding protein C. Those are by far the most common mutations that we see in hypertrophic cardiomyopathy, but we also see mutations occurring with troponin T, troponin C, and troponin I. Exactly how those mutations result in all of the phenotypic manifestations of cardiomyopathy, that's a little bit fuzzy. And here's one author's idea, is that the defective sarcomere alters the amount of calcium signaling, and that then leads to the fibrosis, the disarray, the arrhythmias, and the cardiac hypertrophy. You know, these patients, they get cardiac hypertrophy that then leads to congestive heart failure, but they also are frequently dying of arrhythmias, sudden cardiac death. So in this particular patient, you know, we suspect that he has a mutation in one of these genes. And so we follow the pipeline that we talked about 2 weeks ago. We perform next generation sequencing on that patient, and then we go through the filtering algorithm that we went through before, where we initially identified 4 million variants, but then we threw out some that are frequent. One of the things we also did as we were filtering variants was we threw out those that were outside of exons. And so we're only looking at exon variants, and we found some variants. We actually found two variants in this patient. We found one variant in the troponin T gene, but in public databases, this variant was deemed benign, and so per this particular lab's protocol, it was not reported back to the clinician. And we also found there was a variant in the troponin I gene, but per public databases, this was of uncertain significance. And so again, the lab did not report this one back to the patient. It turns out this patient also happens to have another variant, but it's in the intron segment of the gene. It also happens to be of uncertain significance. So this variant was missed. It was filtered out. It was filtered out because it wasn't located within or near an exon. So the lab never even focused on this variant. So in this particular patient, we ultimately wound up saying our conclusion at the time of analysis was presumed familial hypertrophic cardiomyopathy, but the specific pathogenic mutation remains unknown. Okay, and that's kind of where we were with this patient. And I'm going to digress now to do our recap. And then after our recap, I'm going to come back to this case because really what I want to do is a reanalysis scenario, because a year goes by and some of these variants have changed. This variant, for instance, we had thought that it was of uncertain significance. We now know it to be pathogenic. And so I want to walk through the reanalysis scenarios with you guys because I think they're fairly complex, and because they're complex, they help illustrate a broad range of informatics considerations. So let's turn now to our recap. And again, you know, I think this class could go shorter than usual because for the recap, most of the stuff we're going to talk about, we've already talked about in other sessions. And so I'm going to just kind of mention it without a lot of detail. I think the deck is going to be valuable as a resource to you guys, but I don't know that I want to dwell on it too much because we've already talked about it. And we'll look at, first, clinical genomic resources. And personally, I believe that these are the two resources that have the greatest likelihood today of being actually relevant to clinicians. So there's boatloads of genomics resources out there, informatics resources, bioinformatics resources, but these two in particular are really focusing on determining actionable associations. ClinGen is focusing on disease gene actionable associations, and PharmGKB and its sister organization, CPIC, is really focusing on drug gene actionable interactions. And so the focus of these particular sites is, you know, is this gene associated with disease, and is the variant causative, and is the information actionable? And so, and it comes up with evidence-based recommendations. It comes up with evidence-based recommendations. And likewise for PharmGKB, you can, for instance, plug in a drug and plug in a variant, and you can get back actionable interactions. So I think these are probably the most relevant to clinicians today. PharmGKB is far more mature. ClinGen is getting there, and I suspect over the next 1 to 2 years will become a much more valuable resource. Okay. And looking at this slide, now you get a better idea of why I'm saying I'm not going to walk through all of these in detail. This is my bookmark file. And by the way, when we get down to references, this bookmark file, I stuck it on the download site, and I'll give you the link to that in a little bit. We've covered most of these resources. And if there's specific questions about some of them, then we should talk about that. But I don't really think I want to walk through all of these resources again today. But I call this a recap plus plus because there's a few resources here that we haven't really covered, some that we haven't talked about at all, some that we've really just basically talked about. And so there's just a few resources here that I thought would be worth spending a minute to point out to you. One is the American College of Medical Genetics and Genomics site, and that's probably a very good site to go to if you're looking for clinical guidelines. This is where we get guidelines for determining the quality, the clinical quality of next generation sequencing results, or this is where we can go to get guidelines on how do you interpret a variant to say whether or not it's pathogenic or not. So very clinically focused genomics guidelines. The other thing that's here that we haven't covered in a lot of detail are the bioinformatic and the research tools. You know, we've really tried to focus more on the informatics side rather than the bioinformatics side because the focus of this class isn't for those people that are going to be going off and doing research, but there are some bioinformatics research tools here. The BioStars Forum is for your most nerdy questions about bioinformatics. The BioStar Handbook is a bioinformatics book. You have tools like Galaxy and the UC Santa Cruz table browser. You have tools like mygene.info and myvariant.info, and you have the Center of Excellence in Big Data Computing, an NIH, part of the NIH Big Data initiative. And really what these tools and data sources focus on are more the bioinformatics research type tools. And pretty much, I think that's all I want to say about this page. Oh, sorry, GA4GH. We haven't talked about, but we will be covering GA4GH today. So that's relevant. We haven't talked about it, but we're going to be talking about that in a couple of minutes. So let's see. When we talked about cancer, we focused primarily on the My Cancer Genome site because that was just the site for the example, but all of these are complementary, and they're all, well, at least many of these are working towards this same notion of actionable somatic variants, whereas some of these, like the Cancer Genome Atlas and the Jackson Laboratory Clinical Knowledge Base and COSMIC, are their focus more is on bioinformatics and research, you know, collating all of the different mutations that are seen, whereas things like OncoKB and My Cancer Genome, their focus is more clinical, helping clinicians interpret what to do when their particular patients have different somatic variations. So it's kind of a blend here. So any questions about any of these? Okay, you can have hours playing around with these. One of the other bioinformatics resources that we have spent some time on throughout the course are these HL7 version 2 standards. And as we talked about, there really are two main HL7 version 2 standards. One is the cytogenetic standard, which is used for sending karyotypes, FISH, and chromosomal microarray data. And the other one, which went to ballot in May of 2017, and it hasn't finished yet, is the guide for molecular reporting of genomic results. We looked at communicating discrete genetic variant panels. We looked at that one quite a bit. We looked at pharmacogenomics a little bit when we did our gene testing. We have not looked at section 3 or the glossary. And really, what these other two sections get at is sometimes we know that a complex genetic variant is associated with a phenotype. So for instance, it might be you need to have a variant here and a variant here. Taken alone, just this variant by itself is not pathogenic, and taken alone, this other variant here is not pathogenic. But when you have both of those variants, then together, that's associated with a particular phenotype. And so that complex variant can be communicated here in section 3, and then the discrete component genetic variants can be nested underneath that. These discrete components are just exactly the same representation as the discrete components from section 2. The other thing we hinted at during pharmacogenomics, but it's a tricky subject that we didn't cover in a lot of detail, was that oftentimes in pharmacogenomics, what's being communicated is not the HGDS syntax for a particular variation, but rather what's being communicated is an identifier for the allele of a particular gene. So let's say you have a cytochrome P450 gene. There's multiple different alleles. Some of those alleles are named. There's multiple variations that contribute to one allele, but it's the allele that has been associated with putting you at risk of a drug reaction or making you an ultra-fast metabolizer or a slow metabolizer. So in some ways, the pharmacogenomics is similar to the complex genetic variant in that what's usually being reported here in section 4 is an allele rather than a discrete variant. And just like we have a nested way to communicate the discrete component variants in section 3, here in section 4, there's a way for labs to include the discrete genetic variants that together add up to the particular allele that was associated with the in section 4. Okay, and that's about all I want to say about version 2, unless there's any questions. Okay. Maybe I should say one other thing. One of the things that this particular implementation guide excludes from scope is the communication of simple genetic test results where there is what I might call an atomic level LOINC code. There might be a LOINC code for is this particular variant present? And the answer might be yes or no. And so I point that out because when we start thinking about decision support, it's going to be important that we can recognize the redundant or the multiple ways that alleles can be represented in electronic health records. And so one day we may be seeing a lot of these molecular reporting messages flowing and the cytogenics reports flowing. But just keep in mind that we already are getting results back in some cases using existing HL7 messages where you have a LOINC code that is a question: is this variant present? And the answer is yes, it's present, or no, it's not present.
Yeah. So this is I'd like to chime in here, Bob, because it's Diane Carter, and as many of you know, I work very heavily with laboratory interfaces, and what we see so often, even sometimes with labs like Mayo Medical Laboratory, they happen to be very involved with LOINC, as of ARUP. ARUP is always represented at LOINC, and yet there's a tendency to just send out that result as a big blob of text that has some information about the specific findings and some information about the interpretation, all as a blob of text. So I guess, since you're so active in HL7 standards, there's this kind of broken link between, and we do have the ONC, we have the certification for software standards, but somewhere the link of actually getting laboratories to send out discrete data is broken in spite of the standards recommendations. What do you have any comments about that?
Well, I tend to believe that part of the reason has been the lack of a robust data communication standard, and at least my understanding from Clem is that the feedback he's getting from the labs is, if you build this standard, we will migrate over to it. And if you look historically, prior to this molecular reporting standard, it was somewhat challenging for people to go into LOINC and to know what codes to use. And before there were well-defined LOINC panels, people would be sending things very differently. And so while I know it's not necessarily sufficient, it does seem as though one of the necessary roadblocks that had to be dealt with was, we had to come up with a good standard. And once we have a good standard, then there's a number of ways to bring people along to this. For instance, meaningful use certification, or a lot of different carrot and stick type methods. So I think that this is incredibly important work, what the LOINC committee and what the Clinical Genomics Committee is doing, putting this standard forward. And I think there's going to be various drivers that will ultimately bring labs along towards adopting it.
Right, and I certainly have seen the influence of meaningful use, because hey, it is a requirement, and it has had some carrot and stick associated with it. But I have seen organizations that actually add the LOINC codes to all of their orders and results, but then their foreign interfaces with outside labs, or they do not have that they're interfaced with other organizations that do not do that, and so therefore, how can they tie in their LOINC mapping to result streams that are not mapped to LOINC? So, or to HL7 formats that are used in certain interfaces that do not include that. So yeah, it's quite a challenge.
Yeah. Okay, so that's version 2. And now I'm going to turn to a couple of other HL7 standards. First is this domain analysis model that was published in February of 2017. And it's quite good. It's not a UML diagram of the domain, but rather it's a series of scenarios and workflow diagrams. And it's pretty broad and encompassing with respect to scenarios. And the Clinical Genomics Committee has plans on updating that. And in fact, when we talk about reanalysis scenarios later, there's some information about workflow for reanalysis scenarios in this domain analysis model. And then we've also talked about FHIR. And just as a reminder, there's some FHIR examples from our first session and our second session on the download site if you want to kind of refresh yourself on. I think one was for thalassemia, and I think one was for sickle cell anemia. There was also some HL7 version 2 corresponding examples. And what this picture here illustrates, and this is just taken from the FHIR genomics documentation, are the various resources, but also I think what's interesting here is that it kind of gives you a sense of size. And so when you do next generation sequencing, you may recall we said those files are humongous, on the order of terabytes. And FHIR doesn't deal with files of that magnitude. There's another standard called GA4GH that deals with these massive data files, the VCF files that we've talked about, the FASTQ files that we've talked about. We're going to look at that. We're going to look at those in a minute. But really what FHIR focuses on are smaller resources, the sequence resource that has information about a sequence, and there's a couple of examples here of sequence resources, and then the observation, which is clinically relevant observations about the sequence. Okay, so we might have a sequence resource that says here's a stretch of DNA, and we see 12 different variants on that particular stretch of DNA. And that might be something that's encoded using a FHIR sequence resource. But now, if you look at that sequence and the variants on that sequence, you might say, well, these two in particular we believe are clinically relevant. So we're going to create genetics observations for each one of those clinically relevant variants. We'll point back to the sequence resource, but our goal with the genetics observation resource is to really pull out those variants that are clinically relevant. Okay, and then of course there's a family member history resource that we've looked at before, and then of course this can all be bundled up into a diagnostic report resource that can be triggered by a diagnostic request. And so one of the things that we see here, again, are the observations are like bite size. You know, you can think of a blank code and an answer with a reference to the DNA sequence. The sequence resource is the next size up, but the sequence resource really is a subset of the complete data set that might have come from next generation sequencing. And so I want to introduce you a little bit to GA4GH, because I believe that when we start examining the reanalysis scenario, imagine the case where a patient has their complete DNA sequence and it's now sitting over here in a repository. That's the big terabyte file, and there's no FHIR specification for pinging that humongous database. Right? FHIR deals with sequence resources that are bite-sized, and it deals with the more atomic level observation resources. So if we want to ping a repository or query a repository that's holding the massive amounts of data, such as the raw variant data, the 4 million variants that we saw in our patient with hypertrophic cardiomyopathy, if we really want access to all 4 million of those variants, FHIR doesn't work. FHIR wasn't intended for that purpose. Maybe it will evolve to that purpose down the road, but for now, the predominant standard for pinging these massive databases is GA4GH. Oh, so before I move on, any questions about FHIR resources? Okay. So GA4GH, it's a large organization. It's a highly influential organization. It's a not-for-profit organization. There's over 200 institutions collaborating together, and the data working group of the Global Alliance for Genomics and Health, the data working group said, you know what, we don't have really a standard way to access next generation sequencing data. Illumina has their API, or 23andMe has their API, or this other next generation sequencing platform has their API, but variants are all represented differently in each one of these platforms, and et cetera, et cetera. What if we could come up with a common data model and an API? And this API would allow you to feed this repository. Through this API, you can send FASTQ data into the repository. You can send VCF files into this data repository. So there's a GA4GH repository, and there's a GA4GH API for loading the repository and for querying it. And what's actually helped propel GA4GH into the limelight recently is Google's large-scale implementation of the GA4GH API in the cloud. And I included a reference to that in the slide deck, by the way, the Google implementation. And so Google is actively involved in helping to push the envelope of the GA4GH API. So again, FHIR wasn't designed, and at this point doesn't have the functionality to query the raw data from next generation sequencing. And the idea is that FHIR and GA4GH have this kind of shared sense of boundary. Where do we use GA4GH and where do we use FHIR? And so now we begin to see, okay, there's HL7 version 2 cytogenetics. There's HL7 version 2 molecular biology. There's FHIR. There's GA4GH. And I think, at least for me, when I start to think about it, I begin to wonder, well, wait a minute, where do all of these standards fit, and which one do I use for what? And that's kind of sort of what I want to return back to when we return back to our hypertrophic cardiomyopathy case, because I think the reanalysis scenario is going to help illustrate how all of these standards can be leveraged together in a complementary fashion, in this case, to meet a fairly complex scenario. Okay? So, this is Recap++. This is, I guess, the plus plus. We haven't talked about any of this stuff, and I didn't really intend to spend much time on this. I just wanted it to be in the deck so that you'd have it for reference. These are the large-scale genomics EHR integration projects. Some of these have been going on for several years. Some of them have only recently started. Sync4Genes is relatively new. Ignite, I think, has been going on since 2014. Digitize Action Collaborative, I think they're actually now exploring moving into HL7. Some of these projects, like MedSeq, have resulted in well over 50 publications in the literature. And so what I did on this slide was, here's the URLs for these projects. These are the main ones. These are the main genomics EHR integration projects. And I just kind of sort of, at a high level, tried to summarize for you what some of the major activities are. As you can imagine, when you've had these groups working for several years, there's so much more that these groups are doing than what I could summarize in just a brief list of major activities. And so I would say, when you start to think about your own genomics EHR integration. These are really good places to look. They have a lot of information about hurdles, about barriers, about challenges, about solutions. And you can kind of sort of look at these major activities possibly to say, oh, well, emerged, they're actually focusing on info button. Okay, why don't I start there if I want to implement an info button solution in my EHR? Or, gee, I'm really interested in pharmacogenomics. Well, look, Ignite has studied pharmacogenomics, and Digitize has studied pharmacogenomics. So maybe we should begin by looking at some of their publications. And that's kind of sort of all I want to say about these particular resources. Mainly, I just wanted them here in this deck so that you had kind of a complete list of resources for down the road should you need them. So any questions? Yeah.
This is great. Do you know if there's any major disagreements or conflicting recommendations within these organizations? I mean, not within them, but between each organization. If you would go to one area and look for some recommendations on the family history or oncology, are you going to find some different significant differences?
Not that I know of. I mean, I'm not intimately familiar with all of the work that they're doing. I wouldn't be surprised, but I just don't know.
Okay. So their focuses are, or their goals within the space are different, maybe.
Well, like if you look at Sync for Genes, for instance, they had to, and this is just recently, so I'm familiar with this. They've done, and a lot of these, what these are NIH-sponsored pilot projects. Okay, so there were 5 pilot projects in Sync for Genes under these 5 domains, but ultimately the Sync for Genes group had to get together and formulate a set of specific recommendations back to HL7 for how to modify the FHIR genomics resources. So ultimately, Sync for Genes did come up with, here is our concrete set of recommendations back to HL7. In terms of what kind of rumblings may have gone on to formulate that and what kind of debates may have gone on to formulate that, that I can't say. And we know that, for instance, the MedSeq project has provided feedback for pharmacogenomics, and what's the other one? I don't remember which project this was now, but a lot of these projects, because these are pilots, at least from what I've seen, and it's almost as though genetics is doing it right compared to a lot of other domains that weren't. It's what are these guys doing when they're studying info button implementation and they find that it's not exactly working the way they want for pharmacogenomics? What they're doing is they're compiling a set of recommendations, and then they're working with PharmGKB to kind of implement their recommendations. So mainly what I see here is not a lot of infighting. What I see here is these groups are coming up with fairly concrete recommendations that they then seem to be channeling to the body that can help implement their recommendations. Even though there's probably still a lot of disagreements within, that's just how some people are. Let's see, any other questions on this? Okay. This is also a noisy slide with tons of text that I don't think I want to walk through in a lot of detail. What I did here was I went through all of the clinical scenarios we've covered every week, where each scenario we've identified some kind of bioinformatics challenge or issue. And I've just tried to pull them all together in one slide. Okay, and most of these should be familiar. Maybe I'll just jam through them pretty quick. If there's specific ones that you want to stop and talk about, we can do that. Searching is an issue. You can't search all sites by HGVS. No site could be searched by that other cytogenic syntax, ISEN. And not all sites are searchable by EHR terminologies. And this is recognized, at least by some, PharmGKB recognizes that it's not currently searchable by RxNorm, but they've publicly stated their intent to go in that direction. So searching has been a challenge, but it's a well-recognized challenge. The actual data that's stored within the public knowledge bases, or the data as it's represented in the EHR, also has some issues. There's multiple allele representations. This one here is a little bit tricky. We talked about it briefly in our pharmacogenomics discussion. There's no definitive source of truth for star alleles to HGVS mapping. So two different groups might say, hey, I looked at this particular P450 gene and I found allele number 3. But the actual atomic level, HGVS level variance underlying this lab's notion of allele number 3 versus this lab's notion of allele number 3 can vary. And so that's also a well-recognized challenge, is that there's no, when a lab tells you, hey, I found allele number 3 in this P450 gene, knowing exactly what the corresponding HGVS variants are, and there's differences across labs. Biomarkers, the standardization is less mature, and of course we have still big problems with large structural variants in how they're coded with HGVS and how they're coded with ISCN, and for searching for them. When we look at public knowledge bases, we'll see that some of the knowledge bases don't even include EHR terminology in their knowledge base, so they can't possibly even enable searching by those codes or responding back with those codes. We've seen examples where the SNOMED mappings are not always the best match. There's no existing HGVS to SNOMED mapping, at least that I'm aware of. Each one of these knowledge bases, some of them are beginning to think in terms of a FHIR API, but as it stands, there's no common API to ping knowledge bases. For some of the more actionable knowledge bases, like ClinGen and PharmGKB, not all of the recommendations are structured. And so we're not to the point where we can just do out-of-the-box dynamic clinical decision support based on having an EHR ping these public knowledge sources. And of course, there's no single all-inclusive site for variance, which is something that we came to realize just through the homework assignment, for instance. If you look at variant analysis pipelines, and a variant analysis pipeline includes identifying that a variant is present, as well as determining the clinical significance of that variant, or identifying that a variant is present, and then as time goes by, rechecking that variant to determine whether or not it possibly was initially thought to be benign, but is now known to be pathogenic. So when we look at these pipelines, we see lack of concordance. The data files are enormous. Variant calling is computationally intensive, and we see conflicting pathogenicity reports with evolving categorizations. So not only do they conflict, but they're changing frequently. Which is good on the one hand, because as we amass more and more data, we're getting closer and closer to what's real, what's true. But this is one of the things that is challenging to us, because it means we have to think about a reanalysis pipeline as well as just an analysis pipeline. And then finally, with respect to interoperability, the communication standards I think they're really getting darn good, but they're still maturing. The V2 LRI standard, it's invalid. It's not done yet. And the FIRE DSTU or STU3, that still needs to be validated. The sequence resource still needs to be validated, so they're still maturing. And when we look at SNOMED, it's inconsistent in its approach to creating codes for heterozygote and homozygote state. For some diseases, there'll be a SNOMED code for carrier state, and there'll be a code for the homozygote state, but it seems to be fairly inconsistent. So again, these are, yeah.
I'm sorry, this is Diane again. So I noticed that, in your data representation, these are all standards and you're not talking about ICD. But I was at a personalized medicine symposium, and one of the speakers from Australia was really chiding the U.S. for not moving more aggressively toward ICD-11 and not participating more in the development, because in his view, you really had to go to ICD-11 before you could accurately represent many genetically related diagnoses. Do you have any thoughts about that?
I'm not sure about what ICD-11. It's because of the collaborative work that Chris Schutt's been trying to do between ICD-11 and SNOMED. I don't know if it's really any different than what we have right here. Is it there's no existing mapping, so a lab can send you an HGVS variant, and the thing is, how are you going to align that with a SNOMED code or an ICD-11 code so that you can then be plugged into this whole other set of knowledge resources? And so, and if you work through HGVS, and from HGVS you get into ClinVar, and from ClinVar you can then follow a link to OMIM, and from OMIM you can look at some of the SNOMED codes. And OMIM also has ICD, at least ICD-9 codes. I don't know if they have ICD-10 codes now. When you traverse that entire pathway to get to a SNOMED code, you wind up getting a lot of fuzziness, and it's not exactly a very robust process. So to me, one of the things is, we have the EHR terminology standards kind of over here on the right, and we have the bioinformatics standards over here on the left, and the bridge between them is being developed, but it's not fully there yet. And if we wanted to talk about ICD-11, I think we can talk about it kind of similar to how we think about SNOMED, which is, where's the bridge and what do we want the bridge to be? And in my mind, one place where there might be a bridge is, given an HGVS code, what's the corresponding SNOMED code?
Okay.
I don't know if it's relevant, but I must say when I was working a few years ago at the Permanente Federation, I was pushing for us to have some involvement in ICD-11, and I didn't get any response or interest.
Yeah. So, and again, these are challenges that we've encountered through the course of this class. There's a lot of literature out there on challenges that people have encountered trying to integrate genomics into their EHR. And most of it's the same, but just again to point out, these are the challenges that we have now firsthand experience with, because we bumped up against these challenges through our scenarios. Okay. Okay, so let's go back to our case. Let's go back to our case and see how all of these bioinformatic resources can come together in a complementary fashion to address what I believe to be a fairly complex scenario. And if you remember, we have our patient with hypertrophic cardiomyopathy, and our conclusion at the time of analysis was presumed familial hypertrophic cardiomyopathy, but specific pathogenic mutation unknown. There was a variant found that was benign, so it wasn't reported. There was a variant found that was of uncertain significance that wasn't reported. And then there was a variant missed because of filtering. Okay. So I want to now turn to this reanalysis scenario. I think I want to kind of sort of make a case for it, try to make a case for at least why I believe it's important. And also, I think, as you'll see, it's complex enough that you'll begin to get a sense of how all of these standards kind of work together. So here's some advances over the past year, and okay, there's a little caveat. I kind of tweaked this a little bit for the sake of example. But let's say, for instance, that the variant pathogenicity interpretations have changed. So 2 of the variants that were of uncertain significance are now known to be pathogenic. One of the variants we actually detected in this patient, but this other variant, which is in an intron, we hadn't even identified because it had been filtered out. Now we believe that both of these variants are pathogenic. Also, over the past year, the American College of Medical Genetics and Genomics has, updated their list of variants to report as secondary findings. You may recall that when labs do next generation whole genome sequencing, they report back targeted results based on what was ordered or what the ordering clinician was looking for, but there's also a list of variants that if they happen to find them incidentally, the ACMG recommends that they also be reported. And so, and there's a number of hypertrophic cardiomyopathy variants that are on the ACMG list. And so now this particular variant has been added to the list. We've also learned in the past year that there's another gene. We didn't even know that this gene had anything whatsoever to do with hypertrophic cardiomyopathy. It's kind of like example 4 from the homework assignment. We now know that this is a polymorphic gene. It has polymorphic alleles, and we know that one of them is associated with an increased risk of sudden death and severity of hypertrophy. So there's this thing called the insertion-deletion polymorphism in the ACE gene. And if you're homozygous for the deletion variant and you have hypertrophic cardiomyopathy, that can greatly increase your risk of sudden death, and it will also increase the severity of hypertrophy to the point that it's controversial, but some believe that in the presence of hypertrophic cardiomyopathy and this polymorphism, that tips the scale towards implantation of an ICD, an implantable cardiodefibrillator. We also have learned that there's a gene, the SLCO1B1 gene, that particular variants of that gene are associated with an increased risk of simvastatin toxicity. So it's been a busy year with a lot of advances in genomics, and all of this data that's being accumulated and crunched has really led to some nice advantages. And we have our patient who had whole genome sequencing a year ago. And now, faced with these advances, there's at least 4 reanalysis scenarios that come to mind. Let's go back and look and see, out of all of his variants, does he in fact now have variants that would be deemed pathogenic hypertrophic cardiomyopathy variants? Does he have any variants that are on the ACMG list, that weren't at the time? what is the genotype of his ACE gene? Does he have the homozygous deletion variant that puts him at increased risk of sudden cardiac death? And let's say I want to treat this guy with simvastatin. Well, wouldn't it be nice, since I already have his DNA, if I could just determine the genotype of his SLCO1B1 gene prior to initiation of the simvastatin? And I thought it would be helpful also to point out the clinical rationale for reanalysis. Why would we do this? Why would we bother reanalyzing? Why do we care if, why do we care whether or not this guy is known to have a pathogenic mutation for hypertrophic cardiomyopathy? What are we going to do differently about it? So some of the things that we might do differently, if we know whether or not he's got a pathogenic mutation, is it might help us with family counseling. It might help us determine how aggressively we need to screen his offspring, or it might be something that he wants to use with his wife for family planning. It might be important for screening the patient. There might be different screening tests that we're going to do on him if we know what his variants are. And then possibly most important, it's going to have direct treatment implications on this particular patient. It might tip the scale towards wanting us to place an ICD, an implantable cardiodefibrillator, and also it might affect the simvastatin dose we give him. So when we look at these reanalysis scenarios and we look at all of the different standards we've talked about, what I want to try to do now is give you an example of how we might put together this entire kind of like reanalysis ecosystem. And take this with a big grain of salt. I am in no way trying to assert that this is the infrastructure. This is really just an example reanalysis infrastructure to kind of begin to give you a sense of how all of these different standards might be leveraged together to meet a fairly complex use case.
So Bob, this is Diane, and excuse me for interrupting you, but I really like this topic. I've been kind of poking around, looking at some sample next generation sequencing reports from Mayo laboratories, and they do have an entire section on some of their sample abnormal reports. It's VUS, variant of unknown significance. So that is permanently part of that patient record, but the concept of you could reanalyze that in the future using as the knowledge expands is very interesting. So I'm very interested to hear about your process that you're trying to recommend.
Yeah, and you know what, it was, and honestly, it's the literature is pretty much in agreement that there is a reanalysis scenario that's real and necessary. The literature is pretty sparse on, okay, but then what do we do? And how do we do it? And in addition to showing you an example of reanalysis infrastructure, I want to, after we look at the infrastructure, I want to kind of talk about some of the challenges. This to me is a very complex scenario, and I think that has a lot to do with why no one is publishing solutions, right? And so, again, this isn't a solution because there's a lot of things you have to think about, and I really don't think there's a one-size-fits-all solution. To take this as an example, to me, what's a real-world problem, and we'll delve into this in a little bit more detail over the next few slides. So here, imagine we have a genomics reanalysis service. And this service, or actually let's start here, we have an EHR. And an EHR communicates with the lab, as it always has, with HL7 version 2 orders and results. But when the lab performs next generation sequencing, it's certainly not sending back all of the results to the EHR. And in this scenario, the lab stores the full set of results, all of the, the FASTQ file or the VCF file or what have you, in a GA4GH repository. So for instance, let's say the lab takes the VCF file and uploads it into a GA4GH repository through a GA4GH API. Okay. Now we have a genomics reanalysis service that can support various use cases, and we'll look at the various use cases with a workflow diagram on the next slide. But this genomics reanalysis service can talk to the EHR through FHIR and FHIR Genomics. The reanalysis service can talk to public knowledge sources, and again, there today is no common API for talking to public knowledge sources, and so here I just said custom, for instance, a knowledge base API. Most of the large NIH knowledge bases that are out there do have APIs, but there's no common API. And then also the genomics reanalysis service would have the ability to directly interact and query the GA4GH repository using the GA4GH API. So let's see now. This picture here, I want you to take this picture with a big grain of salt. I had a lot of fun putting it together, but every time I look at it, I want to change it. So again, just think of this as an example. Try not to poke too many holes in it. Think of it as a way of trying to educate on the way that different standards might come together for a reanalysis scenario. So I've got 4 different scenarios here, and using the architecture that was shown on the previous slide, this shows how some of these scenarios might be instantiated to support our needs for reanalysis. And in the first one, this is the simple scenario where an order goes to the lab for next generation sequencing. The lab sends back, using the V2 LRI, the discrete observations. Okay? But in this case, the lab is also storing the raw data, or at least a VCF file, in the GA4GH genomics repository. Okay? And so now every time a patient has whole genome sequencing, all of the raw data is being amassed in this large genomics repository. And that sets the stage for reanalysis services. So here's a case where a reanalysis service triggers a reanalysis for pathogenicity. The variants were of unknown significance a year ago, but now they're known to be pathogenic. Or this reanalysis service triggers a reanalysis because ACMG has updated their list. ACMG has now said that you should also report these 3 findings. And so here we have this reanalysis service, which is communicating with public knowledge bases, and the reanalysis service comes to learn that, there's been, sufficient changes in pathogenicity, or there's been updates, or, maybe a year has gone by. I'm going to send a trigger to the EHR, and that's going to prompt here the EHR to send an order to the lab. And again, there's a lot of different workflows you can envision. And in some cases, you could envision a workflow where the EHR doesn't necessarily send the order to the lab, but the reanalysis service directly interacts with the GA4GH repository. But in this case, we're sending the order to the lab because there's no big rush, and we'd rather let the lab go through their normal QA process. The lab requeries the genomic data using their GA4GH API, and then the lab sends results back to the EHR. But there's still a need to compute the delta, right? I mean, this is, the EHR has information about pathogenicity that may have actually reported to the patient a year ago. So the reanalysis service here continues to interact with the EHR to help compute the delta. What's different from a year ago? What do we need to actually notify the provider about? What do we need to actually notify the patient about? Here, the clinician can also manually trigger a reanalysis for genotyping of the ACE gene. So the clinician just happened to read a compelling article and was already on the fence. I kind of sort of think this guy needs an implantable defibrillator, but you know what? If this ACE gene had the particular variant, that's going to tip the scale, and I'm going to go ahead and do it. So I'm going to order this. The lab then does a reanalysis of the next generation sequencing data and reports back the lab result. And there was no new specimen that had to be collected in this particular case because this was a reanalysis scenario. And then finally, I wanted to throw in a scenario where the lab isn't necessarily involved at all. This would be a real-time clinical decision support where I order a drug. I order simvastatin, and that triggers the reanalysis service to check the genotype of the SLCO1B1 gene because the Pharm GKB recommendations don't say that if you don't have that gene on file to go ahead and order it, but they do say that if you do have genotype information on file, you should use it to better inform your prescribing. So in this case, I'm about to order a drug. The reanalysis service communicates with the genomic data through a GA4GH API. Based on the genotype that comes back, if it's interacting, then the reanalysis service will send something back to the EHR to alert the provider about the interacting genotype to possibly not give simvastatin or to alter the dose of simvastatin. So again, take these with a big grain of salt. You could play around with these things, and in no way, shape, or form am I saying these are the right workflows. These are really meant to be example workflows where I wanted to illustrate, number one, the use of the various standards, and number two, I wanted to illustrate how there, I think, are scenarios in reanalysis where we want to have a reanalysis service or a decision support engine being able to directly communicate with the GA4GH data. Okay.
I really love all these thoughts. I mean, it really could add a lot to healthcare. But and I immediately think, with my public health background and familiarity with vaccine registries and tumor registries in pathology, I think, oh well, why can't we just send all those genes to a registry database? Like CDC surveillance data and things like that. Except for that, I can just imagine, in these days of anti-science and anti-government conspiracies, well, people would react to having all their genes in a government database, But
yeah.
That's not. To me, it makes as much sense as having a vaccine registry or a tumor registry.
Yeah, and it's it's who's to say? Some patients will authorize use of their data for public health, and some won't.
Right.
And here,
of course, I think it could be de-identified, and there might be ways, even though some black box somewhere, maybe within the people who release it, that could get back information specific for patients, but de-identified for use by the government.
Yeah, although de-identification of whole genome sequencing data is a little bit tricky because you can just do DNA fingerprinting on it.
Yeah. I plan to monetize my genes, so.
Yeah.
Okay, well, please let me know. Okay, because I have a couple of, I'm a fan of sequencing, and I have a couple of personal genome databases out there. So just let me know what the going price is, please.
Yeah, just be prepared. You're going to need a very big computer for this one.
Yeah. Same scenario used for other things currently in EHRs, such that, we just decided to call in all our patients with a diagnosis of sickle cell. Boom, pull them all out. We've decided all people over 65 need a special vaccine now. Boom, we pull them out and contact them. I think there's a lot of things where the literature changes, and we can then do a query within the EHR to pull out all patients who've had a certain drug, who've meet a certain criteria. It's very similar to that. Is it being done much?
No, but this reanalysis workflow would support those.
Yeah.
We'll talk about the challenges in a minute, but the reanalysis pipelines, going back and querying these massive data stores, it's computationally intensive and it's expensive. But theoretically, if there's some landmark study that says, you know what, we just realized that anyone that has this variant is at sky-high risk of disease X and should be tested, we could go in, we could write a rule in our reanalysis service that could go back and query all of our patients that have genomic data in here and identify those patients, at least that have been tested, who are at risk, and we could notify them and bring them all in and screen them. So that's certainly a scenario that's enabled by this reanalysis service.
I think it'd be broader. You could do anything within the medical record this way, because I think it's critical, in other cases.
Yeah. And I'm not sure if reanalysis is actually a new. It seems like a very natural next step, and we kind of do that and have been doing it for decades, right? Because a proxy for gene print is basically. I mean, we have race, we have gender, we have many many proxies that we use today in society for reanalysis later. Look at all the stuff with slow acetylation based on your race, and these are the race has been reanalyzed many times, and gender has been reanalyzed many times. So I don't think it's something new, right? I mean, it's just basically an extension of that idea. Yeah.
There's a nuance, I think. When you're typically reanalyzing, you're going into the EHR and you're looking at atomic results that have been QA'd and received. When you're reanalyzing genomic data, like here, for instance, you're reanalyzing the ACE gene, or here you're reanalyzing a different gene, and these particular genes were never reported to the EHR. They're just in this massive pool of genetic data that's in the repository. And in some cases, the quality of secondarily assessed genes isn't the same clinical grade quality as those that were reported primarily in response to the order. And so you have to be a little bit careful, if you just computationally go in there and extract the genotype of the SLCO1B1 gene, or just computationally go in there and extract the genotype of the ACE gene, that's not necessarily going to have the same degree of accuracy as, for instance, the reporting of the lab back on one of the hypertrophic cardiomyopathy genes, because labs do QA and sometimes they do confirmatory testing before they report back results.
Oh, I see. So
that's
you
one
have
of the
some
challenges.
uncertainty. Okay.
Yeah.
Because I was thinking if it that's good to know, because I was thinking you're defining basically attributes of a person, that are persistent and won't really change over time. But it sounds like these attributes are approximations that might need refinement later because it may have not been accurate when it was done the first time, right?
Yeah. And it's what's so complex is that every test has its own accuracy, and the accuracy of the test, if you do whole genome sequencing, the accuracy isn't the same across all your DNA.
Mm-hmm.
And in some cases, labs are doing confirmatory testing.
Yeah.
And every machine has its own accuracy, and so that's why I think it's that adds a layer of complexity here.
Yeah.
I
But it sounds like there's still utility, right? Because even though there's an accuracy issue, it at least tells you there's smoke where there may be fire, and a confirmatory test can rule in or rule out that gene, if it's something of high interest, right?
Yeah.
Like in a research study, you'd gather up all the patients that you suspect have that gene, then if it's really important to know that they absolutely have it, you do confirmatory tests on them.
Yeah, and in some cases, if the lab hasn't stored the biospecimen, in some cases the confirmatory test is not purely a bioinformatics issue. It might require going back to the patient and getting another specimen. If you need absolute confirmation on the genotype of this ACE gene, it might be you've gleaned all you can glean from querying the genomic data store, but you need to call the patient back in and actually get another specimen and do a targeted ACE gene test in order to have the same confidence in that result that you'd have from a result on the hypertrophic cardiomyopathy genes, for instance.
That makes sense. Diane, and once again, in spite of Theranos story and everything like that, which if I had been their lab manager, that product would have been out there and it would not have failed because they just did some really stupid QA things. But most even like non-medical sequencing is like 23andMe and Genos are done on CLIA certified labs, and so a lot of they don't just send out results willy-nilly. The accuracy and is very is validated. There's a lot of validation steps in the development of the medical device and also in the reporting, even if it's not reported for medical reasons. So I would say that it's not going to be in my view, a huge variation between, your genome on one machine versus on another. I think that's carefully controlled.
And let me just point you to another resource here.
Now, if you're talking about lab-developed tests, then it can be, homegrown tests by commercial labs. Sometimes it can be a little less accurate, but I would say for the most part, it's very highly regulated.
Yeah. And this is quite a nice document from ACMG, where they address what you were just talking about, Diane. Okay. Their recommendations for lab standards for next generation sequencing. They do talk about resequencing scenarios in here as well, although they don't talk about all of the resequencing scenarios that we've talked about. They're primarily talking about resequencing scenarios where a lab has reported a variant and now information on that variant has changed, and how does the lab update. But they do talk a lot in here about quality of sequencing, and the quality of secondarily assessed variants and the reliability, even in CLIA certified labs. So this is a very nice document, but it's a little bit dated too, and it's difficult to know. Here we are in 2017. Where has accuracy gone? And it's dependent on so many variables that it's very difficult to talk about accuracy just across the board for next generation sequencing. If we look at some reanalysis considerations, and again, there's a lot, and that's why I'm not proposing a solution. I just wanted to have an example to kind of get the creative juices flowing. All of the challenges we've talked about in the prior slide have to be considered. Where and in what format does the lab store raw next-generation sequencing data becomes an issue. What is the quality of this secondarily assessed variance? And this is a very complex issue, and there's, like you said, Diane, there's CLIA issues, there's, technology issues, and so if you have scenarios for secondary assessment that go through the lab and then come back, then you know that you presume that results that are coming back are of sufficient clinical grade quality. But when you want to start talking about these scenarios where you have a decision support application talking to the genomic data and then feeding that back to the clinician, that's where I think these quality of secondarily assessed variance becomes more significant, and it's an important consideration. Given the rapid improvements in technology and bioinformatics, how long until, say, a VCF file should be considered obsolete? There's this naive notion that, hey, we have your DNA and we're going to store it in a database, and for the rest of your life we'll be able to just ping it and have reanalysis. But the fact is, if you had measured and analyzed my DNA 10 years ago, or if you analyze my DNA 10 years from now, there's probably going to be a significant change in the accuracy across the board of the identified variants. What are the costs associated with the various reanalysis scenarios? How will delta results, such as a change in pathogenicity, be reported to the provider and to the patient, and what should trigger various reanalysis scenarios? So it's a fun scenario in that it's complex, and in that it exercises a lot of our standards to hopefully give you an idea of how all of these standards can work together in a complementary fashion. And just, also, it's not as though there's a one-fit-all solution. It's a complex scenario. And I'm going to move on to just kind of quickly showing you some of the references I've compiled for you. But first, let me just pause and say, any additional questions or comments on the reanalysis scenarios? Okay. So just really briefly, I just want you to see what's in here. All of my bookmarks are available on the download site, and that's the file name. And so, have some fun with those. Then, in terms of references, for this particular course, here's where all the slides are, and here's where all the videos are. And this is the same EHR integration projects from the last page. And then this is the HL7 standards. And then finally, all of the figures that we've been going through in this book, that we've been going through in the course, unless otherwise noted, have been drawn from one of these books. And I think that's it. If we have one more minute, I wouldn't mind asking you guys for your thoughts on today's class. What was good about it? What you'd like to see different about it? I thought today we had really nice class participation, and keep that in mind for next week, because we really need class participation next week.
Bob, you've done a nice job telling information, organizing it in an understandable way. Well, in a way that attempts to be understandable. Some of us are still struggling with understanding, but thank you very much for presenting it so nicely.
Oh, thank you, Diane.
Yeah. And Bob, can you hear me okay?
Yes.
Oh, great.
Okay.
Yeah. So I think it, just from feedback that I've gotten offline from different attendees that they really enjoyed the presentation. Your approach to presenting it and the content. It's been very valuable. So I've gotten a lot. Really mostly positive. Pretty much positive. Comments from everybody, and it's just kind of nice to have you back and doing this. I think just from my side, it's not a critique of what's being presented. It's more just kind of what we want to see more. So kind of getting into future discussions around applicability in clinical setting and what are people doing, and getting a little more into depth in that, which I think we'll also end up doing when we, if we end up doing our grant, that'll be kind of fun. But we may want to update folks on work that we are going to be doing as well. Since we're planning to be a part of this and contributing as well.
Right. Okay. Thank you, Jim.
Yeah. Sure.
Any other? Okay. Thank you guys very much. And next week we'll wrap up. Bye now.
Thanks, Bob. Yeah, this is great.